Hello everyone and welcome to this webinar titled Maximizing the Power of Flow Cytometry with Advanced Panel Design. My name is Heike Leyendeckers and I work as the R&D Antibody Portfolio Manager at Miltenyi Biotech. In the first half of this webinar, I will talk to you about recombinant antibodies and why they are the better choice for flow cytometry. During the webinar, I will discuss several topics, which are listed here as agenda points. I will begin by discussing the role of antibodies and the reproducibility crisis in science. Then I will talk about recombinant antibodies and their significance in addressing this crisis. Additionally, I will elaborate on the validation steps undertaken by Miltenyi Biotech to ensure the reliability of our antibodies. All right, before we dive into the nitty-gritty details, let's address the elephant in the room, the reproducibility crisis. This is a hot topic in the scientific community, and it's essential to understand its implications before we move forward. So let's unpack what this crisis is all about and why it's causing such a stir. Let's take a closer look at what's been happening in the scientific community over the past decade. There's been a growing concern, backed by numerous publications and articles, that many of the results we're seeing aren't reproducible by other researchers. And guess what? Antibodies are right at the heart of this issue. Back in 2008, the Human Protein Atlas project published a paper in which they analyzed more than 10,000 internal and external generated antibodies and showed that less than 50% of them were actually binding their target protein specifically. In 2009, David Rim, a pathologist at Yale School of Medicine, discovered that a batch of antibodies designed to detect a newly characterized cancer marker wasn't giving consistent results and thus led to the end of this biomarker development project. Then in 2012, Amgen dropped a bombshell with a study revealing that only 6 out of 53 hallmark papers in cancer research could be replicated by their scientists. Digging deeper, they found that many of the results in these studies were cherry-picked, not reflecting the full spectrum of positive and negative outcomes. This sparked a heated debate in 2014 about the rigor of author guidelines and the need for more transparent reporting. The National Institute of Health even sat down with leading publishers to find a solution. By 2015, the call for change grew louder. Articles in Nature emphasized the importance of antibody validation and championed a new generation of antibodies, recombinant ones defined by their DNA sequence. Amgen continued to shake things up in 2016 with the announcement that they'd be publishing 3 studies that failed to confirm previous findings. In 2018, another study revealed that over 30% of 185 randomly selected hybridomas contained additional productive heavier light chains, leading to compromised antibody properties like specificity and signal strength and signal-to-noise ratio. So, when we piece it all together, it's clear that a significant chunk of the reproducibility crisis stems from unreliable and inconsistent antibodies. The consequences are twofold. Not only are we spreading unreliable scientific data, but we're also wasting vast sums of money in the process. It's estimated that 50% of preclinical research yields irreproducible data. To put this into perspective, conservative estimates suggest that approximately $28 billion is poured into research annually, only to find that it cannot be reproduced. What's more alarming is that a significant chunk of the spending roughly 36%, is attributed to biological reagents and materials like cell lines and antibodies. This translates to a $10 billion annually being funneled into unreliable reagents alone. In recent years, there has been a growing recognition within the scientific community of the reproducibility crisis, prompting structural changes. One notable development is the integration of AI technology to assess the reproducibility of scientific publication. This innovative approach involves assigning confidence scores to publications, providing a quantitative measure of their reproducibility. However, while such advancements are promising, it's essential to acknowledge that achieving more reliable and reproducible results require a comprehensive examination of the reagents used in scientific experiments. Now that we've delved into the reproducibility crisis and its connection to antibodies, let's explore how recombinant antibodies come into play as part of the solution. Let's take a look at the different types of antibodies currently available on the market, along with their associated features. First up, we have polyclonal antibodies. These are derived from the serum of immunized animals and have the advantage of recognizing several epitopes, resulting in a stronger signal. However, due to their origin from immunized animals, they often lack consistency in performance, making them less suitable for applications like flow cytometry, where background noise can be problematic. Moving on to monoclonal or hybridoma antibodies. These antibodies are produced from single clones of immortalized hybridoma cell lines, which are fused with B cells from immunized animals. As a result, they recognize a single epitope, offering higher specificity compared to polyclonal antibodies. Finally, we have recombinant monoclonal antibodies. These antibodies are derived from a defined DNA sequence, which enables them to recognize only one epitope. This superior specificity makes them stand out compared to both polyclonal and monoclonal hybridoma antibodies. Let's walk through a schematic representation illustrating the production processes of hybridoma antibodies and recombinant antibodies. Starting on the left-hand side, we have hybridoma antibodies. The process begins with the immunization of a host animal with a specific antigen. Following immunization, the host animal's B cells are activated, leading to the production of antibodies tailored to the antigen. These B cells are then fused with a myeloma cell line, resulting in hybridoma cells that exhibit stable expression of the antibody. However, it's important to note that these products may contain myeloma cell line-derived light chains, and genetic drift can occur due to prolonged cell culture. As a result, hybridoma antibodies often display variability from batch to batch and may have decreased purity. Now, shifting our focus to the right-hand side, we encounter recombinant antibodies. Unlike hybridoma antibodies, recombinant antibodies are derived from a precisely defined genetic sequence expressed in a controlled expression system. Being genetically engineered, these antibodies encode only one type of heavy and light chain, ensuring exceptional purity. Furthermore, they are manufactured under standardized culture conditions in mammalian cells, resulting in high consistency and purity across different production lots. In summary, while hybridoma antibodies have been a staple in the field, recombinant antibodies offer a more standardized and reliable alternative, characterized by their purity and consistency. Miltenyi Biotech was quick to recognize the advantages of recombinant antibodies, paving the way by introducing the first recombinant antibody for flow cytometry as early as 2012. In 2015, we made a significant breakthrough by introducing the first core markers for antibody panel building. This marked the beginning of an expansion in 2019, where we broadened our portfolio to include application markers specifically designed for flow cytometry. Fast forward to 2024, and we are proud to say that our recombinant antibody portfolio has grown substantially. We now boast over 1,450 REAfinity recombinant antibody clones carefully validated and optimized for flow cytometry. Let me walk you through how we manufacture our REAfinity recombinant antibodies. We start by cloning the antigen binding region and fuse it with a human IgG1 constant region. This fusion creates a chimeric DNA sequence. Next, we utilize a mammalian cell line cultured under standardized conditions to express this chimeric DNA. We employ the same cell line for producing all REAfinity antibodies, ensuring remarkable lot-to-lot consistency. Now let's talk purity. Our state-of-the-art recombinant production method is carefully designed to eliminate any light chain, cell, or serum-derived impurities. This stringent approach guarantees that our antibodies are of the highest quality, free from unwanted contaminants, and ready to deliver reliable results for your research. It's important to note that all our recombinant antibodies come equipped with the same human IgG1 backbone. This stands in stark contrast to classic hybridoma antibodies, which can exhibit a multitude of different Ig backbones. This consistency in the IgG1 backbone across our entire range of recombinant antibodies ensures uniformity and reliability in their performance, simplifying experimental design and interpretation. On the left-hand side, let's take a look at a classic experiment setup where each antibody in the panel boasts a different backbone. This means that for each of these individual backbones, an isotype control needs to be included. As you can imagine, this leads to complex experimental planning and inventory management. Now, shifting our focus to the right-hand side, we observe the scenario with REAfinity recombinant antibodies. Because these antibodies share the same human IgG1 backbone, only one isotype control is required, regardless of the number of antibodies incorporated into the flow cytometry panel. This streamlined approach simplifies experimental planning and greatly eases inventory management tasks. By mutating the Fc region of the human IgG1 Fc, we effectively prevent its binding to Fc gamma receptors. This alteration ensures that the antibodies maintain their specificity for the target antigen without inadvertently interacting with Fc gamma receptors, thereby enhancing their utility in various experimental settings. Under classical conditions with hybridoma antibodies, the Fc receptors present on the surface of immune cells readily bind to the Fc gamma receptor binding regions of antibodies. This interaction is a natural aspect of the immune system, serving to identify and target antibodies attached to infected cells or invading pathogens. However, in the context of flow cytometry antibody staining, this interaction poses a challenge by potentially generating false positive results. To solve this problem, researchers often use blocking reagents. These reagents are incubated with the cells prior to staining with fluorescently labeled antibodies. While this approach can mitigate the problem to some extent, it's far from ideal. One major drawback is the variability in the titers of blocking reagents required for different samples. Additionally, there's no guarantee that these reagents will effectively block all Fc receptors. leading to inconsistent results and potential inaccuracies in data interpretation. That's why a more effective solution lies in utilizing antibodies with a mutated Fc region, effectively preventing any binding with Fc receptors altogether. This ingenious approach eliminates the need for blocking reagents entirely. As a result, researchers can expect minimal to no background signal, streamlining the experimental process and ensuring more accurate and reliable results. Let's take a moment to compare side by side the performance of hybridoma and recombinant antibodies. As you can observe, the mouse monoclonal antibody derived from hybridoma technology exhibits binding and reactivity with various Fc receptors. In contrast, the recombinant REAfinity antibody shows no such interactions with Fc receptors. The stark difference highlights the superiority of recombinant antibodies in minimizing background signal and ensuring specificity in experimental results. Now that we've touched on recombinant antibodies as a promising solution to the reproducibility crisis, let's delve deeper into the validation steps that Miltenyi Biotech implements to ensure the performance and reliability of our antibodies. Our antibody validation process is built upon three key pillars: reproducibility, specificity, and sensitivity. The specific validation steps undertaken are tailored to the intended use of the antibody. We carefully develop protocols for validation experiments, ensuring that the results align with our predefined specifications. Let's start by delving into reproducibility in more detail. When examining antibody reproducibility, we're essentially assessing the ability of an antibody-based assay or experiment to yield consistent and reliable results across multiple trials. Figure A illustrates two instances of hybridoma-generated antibodies, each exhibiting the presence of a second light chain with a molecular weight of approximately 23,500 Dalton, marked with a pink star. Through our analysis, we have identified this as the mouse myeloma MOPC21 light chain 2, stemming from a rearranged kappa variable J gene. Notably, in the left below example, the quantity of the contaminating light chain significantly surpasses that of the productive light chain marked with the green star. It's worth noting that the majority of standard fusion partners utilized for hybridoma generation are all derived from the original MOPC21 murine myeloma cell line. This lineage connection underscores the prevalence of potential contaminating light chains found in hybridoma-generated antibodies. Additionally, we have identified other contaminating light chains in mouse and rat hybridomas with molecular weights of approximately 22,000 Dalton and 24,000 Dalton, although these are not depicted in the figure. Moving to figure B, we observe two examples of recombinant REAfinity antibodies. In stark contrast to the hybridoma-generated antibodies, both recombinant antibodies showcase the absence of contaminating light chains, validating their purity and consistency. That means that mouse hybridoma antibodies may exhibit the presence of multiple detectable light chains, highlighting the heterogeneous nature of these antibodies. In contrast, recombinant antibodies display a clear advantage with only one defined light chain. In our R&D department, we subject all antibody clones to rigorous testing for specificity and sensitivity in flow cytometry. Only the top-performing clones boasting the highest specificity and sensitivity are selected for conjugation to our fluorochromes. During the conjugation process, we employ state-of-the-art chromatographic techniques tailored to each production step of the products. The stringent purification process guarantees that the final product is free from any free fluorochromes, unconjugated antibodies, or activation reagents that could impact antibody sensitivity. Once the final product is obtained, it undergoes extensive testing on multiple primary samples under various conditions to verify its performance. At every stage of the manufacturing process, we conduct rigorous testing to ensure the quality and consistency of our products. This includes testing both the purified antibody and the antibody fluorochrome conjugate. Our testing procedures encompass a range of analytical, biochemical, and cell-based methods, ensuring thorough evaluation of each product. For the final product, we always verify staining performance with cells to confirm functionality. As an example, let's consider a comparison between two manufacturing lots of 4 different CD56 REAfinity antibody fluorochrome conjugates. Using flow cytometry with a max quant analyzer, we compared the staining performance of the two lots using human PBMCs from a single donor. The resulting histogram curves, represented by black and red, demonstrate that the staining performance was nearly identical across both lots. Let's shift our focus to determining antibody specificity. Antibody specificity refers to the ability of an antibody to recognize and bind to a specific target molecule known as an antigen with high selectivity and affinity. Essentially, specificity indicates how precisely an antibody can distinguish between different molecules present in a biological sample. The gold standard method for assessing antibody specificity involves generating a knockout of the target antigen. This approach allows us to confirm whether the antibody's binding is truly specific to the intended target. Following the generation of the knockout cell line, the next step involves staining the cells with the antibody of interest. We thoroughly characterize the stained cells using both flow cytometry and microscopy techniques. This comprehensive analysis allows us to confirm the specificity of the antibody by assessing its binding pattern and distribution within the cells. In addition to knockout validation, we also conduct epitope specificity assays to further validate the performance of our antibodies. This assay is instrumental in determining whether the antibody recognizes the same or a different epitope compared to a competitor antibody. In a typical staining scenario, cells are first incubated with our antibody, which binds to the epitope of interest. This interaction results in a 100% signal, indicating specific binding to the target epitope. In a second scenario, cells are pre-incubated with an excess of purified unconjugated REA antibody before staining with fluorochrome conjugated antibodies of other known clones targeting the same marker. In this setup, a 100% signal suggests no overlap between epitopes, indicating distinct binding sites. Conversely, a 0% signal indicates complete epitope overlap, implying that the antibodies recognize the same epitope. Following each epitope specificity assay, the results of the experiments are summarized in a dedicated table for each individual antibody clone. Based on the observed fluorescence signal patterns, the antibody clones are categorized into distinct groups. Clones that recognize completely overlapping epitopes are identified as ++. Clones that recognize partially overlapping epitopes are denoted as plus. Clones that recognize completely different epitopes are marked as minus. Let us now turn to antibody sensitivity to find out what it entails. Antibody sensitivity refers to the capability of an antibody-based assay to precisely and reliably detect low concentrations of its target antigen. To validate our antibodies for sensitivity, we conduct a performance comparison assay. In this assay, we stain cells from the same origin, for example, PBMC obtained from the same donor, using antibody clones targeting the same antigen from different companies. To ensure optimal staining conditions, we utilize the proposed titers for the respective antibodies from their suppliers. As you can see, staining with recombinant antibodies shows a stronger signal and better separation of cell populations compared to similar competitor products. This increased signal intensity and improved resolution underline the outstanding sensitivity of our antibodies. All the extended validation information can be readily accessed on the product pages of our recombinant antibodies. At the bottom of each page, you will find a dedicated section titled Extended Validation. This section provides a comprehensive overview of all the available data generated by the R&D team to validate the reproducibility, specificity, and sensitivity of our antibodies. Last but certainly not least, I'd like to share some additional information and services that we offer alongside our antibodies. Understanding the significant effort involved in designing and optimizing panels for flow cytometry experiments, we are pleased to offer a valuable resource known as the Miltenyi Biotech Tested Panels Database. Curated and validated by our in-house experts, these panels are designed to streamline your research process. We recognize the importance of sharing our expertise with the scientific community, which is why we make these panels available to researchers like you. Each panel within the database includes a comprehensive list of reagents and materials, optimized staining protocols, and gating strategies. Moreover, we understand that sometimes the perfect antibody or Fluorochrome conjugation may not yet exist for your specific research needs. That's why we offer the custom antibody design service to address this challenge. Whether you require pure unlabeled antibodies or antibodies conjugated with your preferred Fluorochrome, biotin, or HRP, we have the solution for you. Whether you want single antibodies or complete cocktails, we have a solution for you too. Just contact our service team and it will provide you with single antibodies or complete cocktails tailored to your experimental requirements. In conclusion, we have covered the key aspects of recombinant antibodies for flow cytometry and why they represent the optimal solution for ensuring reliable and reproducible results. However, it's essential to recognize that having the right antibody is just the beginning of your flow cytometry experiment. The next critical step is assembling the right combination of antibodies to construct a panel capable of addressing your experimental questions effectively. In the next segment of this webinar, Omar, the co-founder of EasyPanel, will provide further insights into how you can leverage the EasyPanel Builder to create successful and optimized antibody panels for your experiments. Thank you for your attention, and I encourage you to stay tuned for Omar's presentation.
All right. Thank you very much for passing over, and thanks everyone for being here and for your interest in EasyPanel. So I'm sharing the screen here on Miltenny's website, hosting EasyPanel, our automated flow cytometry panel designer, that is accessible via the link here, or even if you just Google Miltenny panel builder, which you should be able to find via the first link as indicated here. And so, as you guys probably mostly know, EasyPanel is an automated flow cytometry panel designer that would hopefully save you a lot of time whenever you're designing any panels on any of the Miltenny flow cytometers, but also other flow cytometers using Miltenny reagents and antibodies. And so, like I said, it's hosted on Miltenny's website. I'll go ahead and start by selecting a cytometer like the MQ16 here. We show you the configuration, the different lasers and channels, and then moving forward. Second page is where we select the species reactivity. I'll go ahead and choose human, for example, and then we choose whether our sample is fixed, yes or no. We're talking about fixation um, um, after the staining, or sorry, rather before or during the staining. If it's fixed after the staining, then you can just say no. And whether your sample is digested or not. Now, these options or these choices will dictate some choices of clones later on in the panel suggestion that we'll see in a couple minutes. And this is some information that was shared with us by Molteni, saying which clones, for example, which antibody clones are susceptible to digestion or not, meaning that during the digestion process, the epitope that is targeted by this antibody clone will be chopped off potentially or will be affected or not. And so we want to make sure that we exclude products that will not detect a specific epitope that will be affected by the digestion or the fixation process. Users can also exclude fluorochromes, as shown here. We can exclude one or multiple fluorochromes. For the sake of time, we generally suggest to leave this by default unspecified, and then you can always get your panel suggestions, see if there are any fluorochromes that you don't like, and you can always navigate back using the top navigation bar shown here and say, hey, I saw this fluorochrome, which I don't prefer personally, and I would like to exclude it, and then move forward again to recompute or redesign the panel after the exclude. All right, page 3, before we get the final page, which is the most interesting page with the panel suggestion, is where we enter the markers. So we start with the viability die, and then here is where you can add the markers one by one, as I'm doing here. For example, CD3, CD4, CD8, CD25, and so on and so forth. As you may have noticed, the expression level of the different markers is pre-entered by default for many different markers based on mining data from the literature. And this is helpful for matching markers to fluorochromes, right? Because one of the rules of panel design, as you may know, is matching highly expressed markers like this one, for example, to dim fluorochromes and vice versa. EasyPanel is all about automation and saving scientists time, and so we didn't want you every time to say, oh, CD3 is highly expressed, CD4 is highly expressed. So we pre-entered it by default, again, based on different data collected from the literature. Having said that, this is not always accurate. And so I wanted to invite you all to review these default expression levels, and if in your case, for whatever reason, any of these markers has a different expression level than the one indicated by default, please feel free to edit it and to overwrite the pre-selected option. CD4, for example, may be downregulated in the case of HIV/AIDS. And so if that's the case, please, if that's the tissue or the sample that you're working on, please feel free to edit these. Um, the fluorochrome can be left empty, in which case the tool will suggest a fluorochrome that will fit your marker and fit the panel to optimize the result, as we'll see in the last page. But a common question that we get is, oh, is EasyPanel a panel design tool from scratch? Meaning if we don't have any panel. You'd be surprised to hear that actually 80 or 85% of the usage of this tool is by users who already have their panels. So if you have already your panel that you currently use and you currently run and you want to compare it versus what the tool thinks is best, please feel free to enter the fluorochromes that you already use. So in this case, say I'm using a CD3 FITC and a CD25 APC, please enter the different fluorochromes. And what the tool will do is that it will QC the result of your panel and it will get a panel quality score. We call it the EasyPanel quality scores that we'll see in a minute on a scale from 0 to 10, kind of judging the potential quality or the theoretical quality of your panel so that you could see how your panel is performing from a theoretical perspective versus what the tool thinks is best. The other use case, besides, designing a panel from scratch, meaning not entering any fluorochromes, or running your already designed panel to kind of see how it compares versus what the tool thinks is best, is entering panel backbone, or what a lot of people call a panel backbone, which is saying, I'm using this product here and then this product here. But then I want to add one or two additional markers to my existing panel, this panel backbone, and I'm wondering which fluorochromes would best fit these markers. And so this is also totally feasible and a use case for EasyPanel. So anyways, whichever use case you're interested in, please enter the markers and potentially the fluorochromes, edit the expression level. I also wanted to show that you could import an Excel file using this template that you can download anytime to your computer, and upload the list of markers and the potential list of fluorochromes using the upload file here rather than entering them one by one. There's also this cool option or functionality that has been requested by a lot of users. If you want to get some requests for, say, so we have an atlas of all human and mouse cell populations, immune cell populations. Say I'm interested in human Th1 CD4 cells, for example. Whatever you select, you'll see a list of cell surface markers, but also secreted cytokines and transcription factors, which you can add or not, and then you can add them automatically to your panel. And this list is suggested based on, again, mining data from the literature. And if you click add, you'll see that this full list will be added. You can remove some markers. You can add some more markers. This is up to you. This is just a suggestion. Similarly, if you're using a kit, so let's say I'm using this Miltenyi, um, I don't know, MODC analysis cocktail kit with this catalog number. This is something that you can also select, and you'd get the list of markers, fluorochromes, and clones, and you can confirm to add them automatically to your panel. Maybe one last point is that if you click Show Advanced Option, Show Advanced Options, you'll see a few functionalities here. For example, selecting clones. Some users want to pre-specify a clone that they are working with, or inversely exclude a clone. And by the way, you can do it for multiple clones if you want. So this is something that has been highly requested and that is definitely supported by EasyPanel. In case some of you guys work on a cross species panel, for example, a humanized mouse, some markers would be human, some others will be mouse. Please feel free to hit the show advanced options and to fine tune the species reactivity for each marker. You can set multiple markers on a dump channel, meaning that they will all be matched to the same fluorochrome. In order to do so, please hit the dump channel option and click add marker. And here I can say, well, I want to add CD4 and CD19 to be matched to the same fluorochrome. And then, as I said initially, you could either let the tool specify that fluorochrome or specify it yourself. You can also play around with the co-expression. So for this purpose, you can say, well, this marker is co-expressed on the same cell population or the same group, which we'll call group one, for example, here as this one, and then as a third one. But then this one is also co-expressed on a second group or second cell population with here, for example. What this will do is that it will match these markers that are co-expressed on the same group to fluorochromes that have low spillover score, and I'll explain more what the spillover score in a minute in the last page that we'll see. Having said that, you don't really need to enter the full co-expression profile because the tool or the algorithm by default tries to select a combination of fluorochromes that minimizes the spillover. And so I'd suggest you really use this functionality, this co-expression feature for markers that you really care about. Typically, the last gating window or the last gating figure, if it's really two or three markers that are really co-expressed on the same population and that you care about. Otherwise, you don't really need to add all the markers as co-expressed on the same population if that's your case, because this is accounted for by default by the tool. All right, let's so now that we have part of our panel, I'll put this for now and I'll move forward. As I said, well, let's actually add a fluorochrome here, and then let's say a fluorochrome here. Actually, and then a fluorochrome here, for example, and I'll add the last one. Um, And if you have any fluorochromes, even from vendors other than Miltenny, we also do support this. We want to make sure that your panel will be QC'd or will be optimized by this tool, regardless of the fluorochromes you're using. So please feel free to select the fluorochromes or to leave them unspecified. And then moving forward here, in a few seconds, maximum 15 to 20 seconds, you'll get a panel suggestion or a panel QC that will be shown as such. So it's a table that is color-coded with different lasers of your cytometer, in this case, the MQ16, showing the lasers, the channels, the different markers you selected. For each marker, there's like a small information icon showing you the expression level, which was predetermined in the previous page. And then if you click, you'll see some more information about the marker, such as what does it bind to, what is it expressed on, what is it not expressed on, what is its biological function, if you're interested in reading more about that marker or that antigen. For each marker, you'll see a matching fluorochrome, which is something that either you selected or was suggested by the tool. If you're the one who specified this, you'll see a star sign. You guys remember that I chose PESci7 for CD130, PESci5, sorry, for CD137 as an example. And I also chose APC for TIM3. But then the others will suggested or optimized automatically by the tool. You'll see also the brightness score on a scale from 0 to 5, which again is important in the matching of the marker to the fluorochrome. Typically, this is a marker that is highly expressed as entered in the previous page was matched with a dim fluorochrome and vice versa. In the very right, you'll see commercial product suggestions for the different products from Miltenny. So if you click the three bars here, a pop-up window will open showing, all the products for Miltenny corresponding to this marker fluorochrome combination, showing the different catalog numbers, what this product reacts to, the clones, whether the clones were published in OMIPs. OMIPs are flow cytometry, highly reviewed publications. Isotype, the size, the concentration, if we have it, the recommended quantity or dilution, the regulatory status of the product, and then the link to Miltenny's page in order for you to be able to purchase that product. So if I click here, you'll be redirected to Miltenny's page selling that specific product, which you could then add to your cart to purchase if you're interested. Alternatively, you can select the product here, select the product there, just as I'm doing. And you'll see the catalog numbers of the products you selected shown here. And then at the end, you can export the result of your panel using the export CSV, which will download an Excel to your local computer, showing really everything that you see here, including the catalog numbers you selected. Alternatively, if you want to, this tool is also integrated with electronic lab notebooks, which means that if you click share, you can say TH1 panel demo. You can call this panel whatever you would like. And then when you click share, a link will be copied will be generated, and then you can copy it and paste it in, as I said, your electronic lab notebook, in an email, in a Teams message, in a manuscript, really in anything. And anyone who clicks on that link, even if they've never heard of EasyPanel or don't have an account, will be able to access your panel and see your result. So we also show the spectrum viewer, which I'm sure you're all aware of, showing you the normalized emission spectra of the different fluorochromes and the different lasers. And then the EasyPanel quality score on a scale from 0 to 10. So there are some question marks right next to a lot of fields for us to explain what this field is or what that means. And so this is, as indicated here, is a quality score on a scale from 0 to 10. 0 is worst, 10 is best. And it kind of judges, again, the theoretical quality of your panel. Reason I'm saying theoretical is that there are limitations behind our algorithm, just like there are limitations behind any algorithm. There are some criteria and some factors that we don't account for because they're really complicated, such as the laser intensities on your flow cytometer, the PMTs, the titration that you've decided to use, some experimental conditions. These are really hard for us to account for. And so, this is why I'm saying this is the theoretical quality of your panel. Other things like the ones I mentioned could impact your panel, but are not taken into account by our algorithm because they're really, really challenging to account for. But if you click on details, you'll see how this EasyPanel quality score is computed. So you'll see here the different criteria, and for each criteria, you'll see your score out of the maximum or the best possible score. And then the total, as I said, should be a score out of 10, from 0 to 10. 0 is worst, 10 is best. A common question that we get is, what is a good score? What is a bad score? So we're very cautious in suggesting what is like an absolutely good panel or a bad panel, because it's not black or white, it's a continuum, right? But I would generally say, based on feedback that we've received from users, generally things that are below like 7.5 or 7 probably have margins for improvement, and things that are above 8 or 8.5 are probably pretty good panels. But again, this should be taken with a grain of salt. And the best thing to do is to always compare your EasyPanel quality score, meaning with the fluorochromes that you chose, like this one and this one, in my case, with the EasyPanel quality score for a panel that would have been fully designed or fully computed by the tool, meaning that had the tool not, not had the tool not pre-selected the fluorochromes that you have pre-selected, it would have designed a fully designed panel that here in this case would have had a score of 10 out of 10, which is better than my panel. And if you wanted to view that panel, you can click view. And this is how you'd move between the two panels: your requested panel with your own fluorochromes versus the EasyPanel fully designed panel, with the fluorochromes that we suggest from scratch. And so this is again how you'd move between the two and how you'd see some potential margins for improvement for your panel. Back to this window, I just maybe wanted to cite one example of the different criteria that we account for this panel design, for this panel quality score, such as, do you have any markers that are dim and that were matched with dim fluorochromes? Because this is not ideal, right? Again, you want to match dim markers with bright fluorochromes. And so if that's the case, you will lose some points. I lost some points here probably because I had some fluorochrome pairs with high spillover or similarity. In my case, I think that was the case for APC and PE Sci 5, I'm assuming. And these are potentially high spillover scores are things that you could observe using the spectrum viewer or also using the theoretical similarity matrix. And what I see here is PE Sci 7 has a relatively high spillover or theoretical spillover into APC. Um, and so this is probably why I lost some points in this criteria. But anyways, even if I screw up my panel again, let's actually put a dim marker. Um, do I have any other dim markers? Let's put this as dim or like low expressed and put it on PRCP. If you cannot select a specific fluorochrome, either because you don't have the laser or the channel will give you an error message, or if that channel is already occupied by another fluorochrome, will also give you this message as you have seen. So I'll put this on PRCP. PRCP is typically a dim fluorochrome, and this is a marker that has a low expression profile. Um, and I'll redesign and what I should expect now is to see my quality score going further down, which is the case here. And you'll see that part of the reason is that I had a marker that was low expressed that was matched with a dim fluorochrome. And so I got a 0 out of 2.6. Not only do we show you this, but we tried to highlight or to pinpoint the issue, saying that CD94 was entered as a low expressed marker and it was matched with PRCP, a dim fluorochrome. And here you can see the comment highlighted by a yellow warning sign saying that this is not great. So hopefully this will be helpful in optimizing your panels or QCing or having a sense of their quality. The last thing that I want to touch on is the possibility to customize or to edit the panel manually. So for this purpose, please click on the green button that I just showed and you'll see here the different markers and fluorochromes that were matched. If you see any matching that you don't like for any reason, that is totally fine. Feel free to edit it. So here we'll show you the configuration of your cytometer, the MQ16 in this case, and we'll show you the commercial availability for each of the different markers in Miltenni's commercial database. And initially, this was on another fluorochrome. Let's say I prefer to put it on PE. That's totally fine. Select PE. You may even select the product by saying, well, I like this, or I would like to choose this catalog number. Similarly, if you don't like PE Bio 770 here, you prefer to put this on APC, that's totally fine. Please feel free to select any fluorochrome or any product that you like. And then going to the bottom, move next, or you may also move next directly through the top navigation bar to recompute your panel and to get your new quality score. Eventually, you may get some gating strategy figures shown in the bottom, showing you suggestions for gating strategy. These would be images copy pasted from open source sources like some publications or references available online that would be helpful even for your data analysis. Last thing I want to say is that we've been around for 3 years. I think the feedback that we've been getting is very, very good from expert flow cytometrists who actually purchased these panels and who tried them and who ran them. So, but please feel free to let us know if you have any questions, any comments. But again, I think, and I'm hoping this tool will save you a lot of time, but will also highly optimize your panels. You can read more about our website, specifically, different testimonials by key opinion leaders in the field of flow cytometry. But also for those going to the Global Flow Cytometry Conference, CYTO, this year or the following one, you'll typically find people with posters who design panels on EasyPanel and who are, showing how they worked. So hopefully this again will be a helpful and time-saving tool for you all. Thank you very much.